Hello, I'm Laura Marshall.
And I'm Justine Murphy, and this is Light Matters for April 30th, 2014.
On this week's show, an optogenetic switch turns neurons off, new lenses turn phones into dermascopes, nanowires absorb and emit light, and plasmonic probes quantify breast cancer gene segments.
Since its inception about a decade ago, optogenetics has been more effective at switching neurons on than off. This is no longer the case. Building on this discovery by Stanford University professor Dr. Carl Dieserath, in which light-activated opsins from algae pass electrical current into neurons to control brain circuitry, a team has re-engineered the light-sensitive proteins to enable more efficient regulation of neuron cells' off switches. The inhibitory and excitatory switches were comparable in power to a squirt gun and a garden hose, respectively, according to Dieserath. To turn channel rhodopsin into an effective inhibitory channel, the researchers needed to create a lining in the central pore. They did this by re-engineering the excitatory channel and changing 9 of its approximately 300 amino acids. Changing a 10th amino acid allowed them to keep the new negative channel open, which made it more like the positive channel and created a more light-sensitive inhibitory channel opsin. With this advance, the researchers expect the opportunities for optogenetics to expand to therapeutic applications such as pain management. The research was published in Science.
High-resolution silicon polymer lenses can now be baked in conventional ovens and attached to smartphones to make inexpensive microscopes. Dr. Wei-Ming Lee, a professor at the Australian National University Research School of Engineering, made the discovery, which involves harvesting solid lenses of varying focal lengths. Creating the lenses is a simple process in which a droplet of polymer is placed onto a microscope cover slip and inverted. Gravity then pulls the droplets into a perfect curvature. Adding small amounts of fluid to them results in a magnifying power of up to 160 times with an imaging resolution of 4 microns. The droplets are cured by baking them at 158 degrees. The material used, polydimethylsiloxane, is also used for contact lenses, and it won't break or scratch. Lee and Dr. Tri Phan of the Garvan Institute of Medical Research also developed a lightweight 3D printable frame to hold the lens on the smartphone, as well as a coin battery and miniature LED lights for illumination. The silicon polymer lens system could enable ordinary people to examine their own skin for signs of disease or help farmers check their crops for pests. It will be particularly useful in developing countries, Lee said, given the low cost to manufacture the lens system and the lack of access to modern technologies in some regions. Each droplet costs about 1 cent to make, and an entire system for one smartphone costs about $2. The work is published in Biomedical Optics Express.
Stretching gallium arsenide nanowires makes them better light emitters, while compression makes them better light absorbers. Researchers at the Norwegian University of Science and Technology and IBM have found that semiconductors made from a hexagonal crystal structure called quartzite exhibit a pseudo-direct bandgap configuration where electron and hole wave functions overlap strongly. However, optical transitions between these states are impaired by symmetry. By inducing a reversible direct to pseudo-direct band structure transition through strain, the luminescence of quartzite gallium arsenide nanowires can be switched on and off. The researchers have been developing ways to grow nanowires from semiconductors and studying their properties since 2010. Over the last 2 years, they have been growing gallium arsenide nanowires on graphene using molecular beam epitaxy. This discovery could enable more efficient LEDs, photodetectors, thin-film solar panels, and other flexible electronic products. The research is published in Nature Communications.
Hyperspectral imaging using plasmonic probes can identify BRCA1, a tumor suppressor gene for breast cancer, within individual cells. Purdue University researchers measured the number of BRCA1 mRNA splice variants in a cell, which can indicate whether the gene is being underexpressed, which is a possible sign of breast cancer. Existing BRCA1 detection methods rely on samples made up of hundreds or thousands of cells and cannot provide detailed information about how genes tied to cancer are being expressed. The new probes are made from gold nanoparticles, or monomers, tagged with oligonucleotides. Upon injection into a living cell, the monomers can hybridize to 3 selected BRCA mRNA splice variants, forming dimers that exhibit distinct spectral shifts due to plasmonic coupling when imaged hyperspectrally. The monomers reflect green light, whereas the dimers reflect red light, and the number of dimers present corresponds to the cell's number of BRCA1 mRNA splice variants. The technique can quantify mRNA splice variants in a single cell in about 30 minutes. It could also increase understanding of cell biology and pave the way for genetic profiling and diagnosis based on a single cell. The research was published in Nature Nanotechnology.
That's it for this edition of Light Matters.
Email us with your questions or comments at lightmatters@photonics.com.
Thanks for watching. We'll see you soon.